The second large process of gene expression is translation. And translation is the process by which a ribosome reads an mRNA molecule from one end to the other, and as it's reading, it uses the nucleotide sequence in the mRNA, or the codons, to create a polypeptide. It fits into our picture of gene expression right here. The mRNA was transcribed from a stretch of several thousand nucleotides in the DNA, and that stretch of several thousand nucleotides was the gene that encoded this polypeptide. Now remember, lots of different types of RNA can be transcribed, but the type of RNA that's going to actually be translated into a protein is mRNA. That mRNA is transcribed in the nucleus, and it's processed in the nucleus before it's exported into the cytoplasm in order for the ribosome to translate. Just to again put it into perspective, remember that a chromosome is several million base pairs long. Now, within that several million base pairs is a stretch called a gene, and one of the two strands of the gene will be serving as a template for the production of the RNA. This RNA is made based on base pairing with the template, and then that RNA carries the sequence of codons, like the codons you see in the genetic code, that then can be interpreted and translated to create our stretch of amino acids, or our polypeptide. Not only is the mRNA important in translation, as it's the thing that's actually getting translated, but also tRNA is important in translation. There are several hundred tRNA genes in the genome of eukaryotes, and many different types of tRNAs. A tRNA functions as an RNA molecule. This molecule is a single-stranded RNA molecule, but it is base paired with itself to create what's called a secondary structure. The base pairing is hydrogen bonding between A and U, or C and G, and all tRNAs have this same basic shape. Notice two important sites of the tRNA. One is the anticodon. The anticodon is going to assist the ribosome in reading a codon. The anticodon will base pair with a codon, and the base pairing is based again on hydrogen bonding, the same base pairing rules that we've seen before. This anticodon actually specifies what amino acid is attached at the second important site, called the amino acid attachment site. For example, the codon UUU encodes the amino acid phenylalanine in the genetic code. The anticodon here would read AAA, at least if this is the tRNA that's going to base pair with UUU. AAA is here, but the amino acid is phenylalanine. It's the amino acid that corresponds to the codon in the mRNA. The anticodon is not the sequence of 3 nucleotides that you'd read for the genetic code. Instead, it's the codon that it's base pairing with that then determines which amino acid is attached here. There are amino acid attaching enzymes that actually recognize the anticodon and the amino acid attachment site. And these enzymes, with the help of ATP, because it's an energy requiring process, attach the correct amino acid to the tRNAs. So you've got 20 different types of these synthetases, or attaching enzymes, out in the cytoplasm, one type for each amino acid. And these 20 different synthetases recognize all the tRNAs that must have the corresponding amino acid attached. So there's a synthetase that would specifically recognize AAA and attach phenylalanine, which is the encoded amino acid by the codon UUU. Also important in translation is the ribosome. The ribosome is this huge factory made up of many proteins. And I say it's a factory because it's the structure that actually assembles and makes the polypeptide. It is made up of two subunits, the large subunit and the small subunit. And in each subunit, there are many different proteins. The proteins that are functioning in the subunits and our RNA, or ribosomal RNA, are responsible for reading codons in mRNA and creating the polypeptide. If you could look at the structure of the ribosome in the middle of translating, you'd notice that there are pockets called the P and the A site. And these pockets are places where tRNAs are found. Now, the type of tRNA that's found there is going to vary depending on what type of codon is being read. But within the structure of the ribosome, there are sites that are sites for the binding of tRNAs, which are, remember, helping the ribosome read the codons that are found on the mRNA. So what I want to do is actually walk through the process of translation initiation, elongation, and termination with you, so you see how the ribosome works with tRNA in order to create this polypeptide based on the codon sequence in mRNA. First, the mRNA has a stretch at its 5' end, where the cap is found, and that stretch is untranslated. It's like a little binding site for the ribosome. Then, just a little bit downstream of that, or away from the 5' end, is the start of the genetic message, and that means the very first codon, which the ribosome will read to start translation. That start codon is always AUG, and that's indicated in the genetic code as well. Then codons follow one after another, no skipping, no overlapping of nucleotides, until a stop codon is present. And then after the stop codon, there's also a little bit of untranslated mRNA, and then, of course, that poly A tail that we looked at when we talked about modification of mRNA. Initiation of translation involves the binding of the small subunit of the ribosome and the binding of the very first initiator tRNA. This one's going to be carrying methionine because on the genetic code, the start codon, AUG, encodes methionine. The ribosome anchors itself here at this untranslated 5' stretch of mRNA, and it and the tRNA scan until this tRNA locks on to the AUG start codon. Once this has hydrogen bonded with the start codon, then the large subunit settles on and translation initiation is done. Here we have a fully assembled ribosome at the exact start codon of the mRNA, and notice that our very first tRNA is in the P site of the ribosome. And the P site stands for peptidyl site, or you can think protein site, because the protein or polypeptide is always extending from the ribosome in this site. What's open is the A site, and the A site is aminoacyl, or think amino acid. This is the site for the incoming tRNA carrying the next amino acid. During elongation of translation, or the actual synthesis of the full polypeptide, there is always this empty A site for an incoming tRNA carrying the correct amino acid. Now actually all the different tRNAs sort of diffuse in and out, and only the one that can actually base pair with the codon sticks and stays. Once it sticks and stays, that actually causes a little shape change in the ribosome, and a peptide bond is formed between the growing polypeptide and the newest amino acid that just entered, being carried by the tRNA. After the peptide bond formation, this bond is actually broken. So the polypeptide has grabbed the new amino acid, this bond is broken, and the ribosome translocates or moves down one codon. When that happens, then this tRNA that was bringing the newest amino acid shifts over to the P site. This one will be ejected. Now we've got an empty A site again, and the growing polypeptide in the P site of the ribosome. And this just keeps happening over and over again until the ribosome finds a stop codon in its A site. A stop codon simply tells the ribosome to disassemble. And that's not shown here, but or how it happens isn't shown here, but there are actually proteins that come in and base pair, or if they don't base pair because they're not nucleic acids, but they hydrogen bond here, and they cause the disruption of this entire unit. So just to summarize the process of gene expression, Transcription results in the production of an mRNA. And transcription is just that production of mRNA from one little stretch of double-stranded DNA, one gene among many along the length of a chromosome. The mRNA enters the cytoplasm and binds with the ribosome, where a polypeptide is created. Not only is the ribosome important, but also the tRNA, which is constantly recycled and rejoined with its correct amino acid, so that there's this constant supply of tRNAs bringing in new amino acids to the ribosome. Once the ribosome has gone through many rounds of elongation, basically as many codons as there are in the mRNA, the ribosome finds itself at the stop codon. Proteins bind in the A site to cause disruption of the ribosome and the polypeptide. The polypeptide is released. The polypeptide will become a fully functional protein once it's properly folded and modified. Once that's occurred, then it's a protein and it can go do its job. I would like you guys to take a minute to push pause on the video and try to fill in the blanks of this flowchart to give you an idea of the overall process, or see how you're feeling about the overall process of gene expression. Once you've done that, push play again and we'll go through the answers together. DNA is copied into RNA by the process of transcription. DNA is made from monomers called nucleotides, and the DNA is transcribed by an enzyme called RNA polymerase. RNA comes in three different kinds: mRNA, rRNA, and tRNA. rRNA is part of the ribosome. tRNA is the molecule that the ribosomes use in order to incorporate new amino acids. These are amino acid bearing molecules. The ribosome is performing the function of translation, which is the creation of a protein based on the codon sequence in RNA. The protein is a polymer made from monomers called amino acids.